Welcome to BIOS Roundtable. Today, we're incredibly excited to welcome back two phenomenal friends of the show in Hansjörg and don.Ingber. Thank you both for joining us. It's our absolute pleasure to host you once again. To help co-host this episode, I'm joined by my colleague, Andrew Yashar. Guys, let's kick things off. Hans, can you share a brief intro with us, please?
Yeah, so I'm Dutch. I trained in Holland. I'm both a biologist and a medical doctor. Never saw patients after I got out of medical school. Decided to be a full-time scientist. Graduated with a Ph.D. in Holland, then did a postdoc in Boston for 4 years in the Dana-Farber in the late 80s when molecular biology slowly became prominent. Moved back as an immunologist, became actually head of a department of immunology, clinical immunology in the University Hospital in Utrecht. My research then took me away into the Wnt pathway, cancer, developmental biology. I moved 10 years later to head the Wyss Institute, which is an ancient developmental biology institute in Utrecht. Did it for 10 years. While having these managerial roles, I always maintained my lab. So I've always had these two roles. I guess like Don combines these things as well. Then I was president of the Royal Academy, a very political, media-heavy thing, which I did for about 3 years. Very interesting. And then I helped, it was the last big thing I did before I came to Roche. I helped build a national pediatric oncology hospital from scratch, essentially. So all Dutch kids with cancer now go to a single center in Utrecht. It's the biggest in Europe and one of the biggest in the world now, and has taken off quite well. Helped by our Queen Maxima, was protected us from all sorts of outside efforts to not let this happen, and I'm very proud that this happened. And then very recently, I moved to Basel, Switzerland, while still keeping an eye on my labs in Utrecht as an advisor now, but to head Roche's R&D efforts.
Don, we'd love to get a quick overview.
Sure. Similar in a way. I'm from New York. I went to Yale undergrad, graduate school, medical school, so I did a combined MD, PhD. Never finished my residency either. Got a postdoc with Judah Folkman at Harvard, came up here, worked on angiogenesis. I should say, though, that probably my driving force behind my work started when I was an undergrad at Yale, where I took a sculpture class the same time I was doing molecular biophysics and biochemistry, and had this aha moment of seeing that cells use a form of architecture called tensegrity that comes out of Buckminster Fuller world. And that led me to really go deep into developmental biology and cancer and see the important role of mechanical forces. That kind of is a theme through my entire career. So when I went to work with Judah Folkman, I pursued cell shape and cytoskeleton, which was cell shape was something he was interested in the context of angiogenesis and cancer. I did work on integrin signaling and showed that integrins mediate mechanosensation. Got involved in the startup world through Bob Langer was part of that department as well. And have stayed on at Harvard for the last 39 years. Last 14, I've been the founding director of the Wyss Institute for Biologically Inspired Engineering. I'm now a chaired professor both in the medical school and the engineering school as well. But the Wyss Institute, big administrative responsibilities for about 400 full-time staff, as well as keeping a lab, just like Hans. And the last 14 years, I've crossed over in many areas, diagnostics, therapeutics, but probably the biggest part of my effort has been in developing human organs on chips, which is what we're going to focus on today.
And that's the perfect segue. I'll now pass it off to my co-host, Drew, to guide us through that first topic. Thank you both.
Absolutely perfect transition here. And thank you once again for joining us. But from each of your podcasts previously covering your work, we were really excited to have you both back on the show to continue this conversation just from another lens, this kind of roundtable setting to really just debate the true future of human models within bio. Before we really dive in here, I would really love to set some context for our audience through your own lenses and perspective. Hans, potentially you could start. Could you tell us more about the evolution of human models within the life sciences sector? Maybe you could divulge the short but storied history from your perspective.
Yeah, so I guess traditionally we all have worked with cell lines, and everybody who works with cell lines know they're usually easy to handle. Yet they, when you sequence them, they essentially are cancer cells, and they resemble to some extent the organ they come from. They can model some of the physiological functions of that organ, but of course they have the limitations. They're heavily selected. They grow in plastic in 2D. We just heard from Don that cells like integrins and extracellular matrix, and that is not what they typically see in a petri dish. So I guess people have attempted things that are closer to human tissues, human organs, like explants, short-term cultures from with real primary cells. The challenge there has always been that it's very hard to keep them alive. And there was sort of a dogma 10, 15 years ago that a normal human cell will not persist in culture. Although we knew some very good examples like embryonic stem cells or T lymphocytes could have been cultured when I was a PhD student. Skin, Howard Green has actually shown how to do that in the 80s and applied it clinically. Yeah, so even in my lab, and when we actually worked on stem cells, to maybe give a little bit of background, in the gut, which are a little bit unusual, is that they're extremely active, they divide constantly. And that was sort of an inspiration to see if we could do something like what people do with ES cells. Get the cells out of the mouse or human body, the stem cells, and then expose them to a set of conditions, growth factors, extracellular matrix type environments to see. And the intention is really to create, for on a single stem cell, create many gut stem cells in this case. And that, much to our surprise, so there was a lot of serendipity in this process. Much to our surprise, actually, they started rebuilding what they were building in a real body is little bits of tissue that resembled the inner parts of the intestinal tract. And that's when we started using the word organoid. Now that word has been used a lot in the past already for organ-like structures grown from cell lines of primary cells. So at the time we were doing this fully independently, there was a second technology originally pioneered in Japan by Sasai, which didn't start from adult stem cells, which is what we do. So our cells are fully specified. They're pretty dumb. They can only make the tissue from which you derive them. These embryonic stem cells or iPS cells, they have the capacity to become anything or everything, and you really have to take them through the journey that a cell normally takes during development. And so that and that sense, so with this other technology, you can make mini brain or cerebral organoids, heart muscle. So we can only make the tissues where there's a strong stem cell presence, most epithelia probably, but we cannot make heart muscle or brain. And that's where this iPS-based technology comes in, and essentially through developing rather than maintaining the tissues in culture. And I guess at the same time, while this was all developing, I guess people from Don's discipline brought in engineering, because our things are pretty, they build what they would want. There's no restraint around them. There's nothing, say, validated or standardized, and that's, I think, where two fields currently are very intensively collaborating to basically have essentially build the right pieces of human organoids in the right context. So that's where the organal chip technology comes in. That would be my perspective, but I guess Don comes in from a slightly different angle. But we seem to be.
I end up in the same place though, so it's good. I mean, for Drew, I mean, I came at this from, well, first of all, I'm an animal lover, and I did a lot of animal work when I was a grad student in cancer. And when I joined Judah Folkman's lab, I decided that I wanted to try to see if I could do as much of this in vitro as possible. And so I developed all sorts of different in vitro model systems. And also, I mentioned that I worked on extracellular matrix and believe that mechanical forces, the idea whether cells stretch or round, dictates their function. I wanted to show that this was true, and so I developed all sorts of in vitro models, which led me in around very late, around 1990, I guess, a collaboration with George Whitesides that had this idea of creating little islands that would be coated with extracellular matrix, but that would be the size of an individual cell, and that it would be surrounded by a non-adhesive Teflon-like region, so that I could make an island that was, big circle, I'd get a cell would spread, because cells spread by binding and pulling themselves out. And when there's no resistance around the outside, they would stop. So I'd get a pancake-shaped cell. If I made a smaller circle, I'd get a cupcake. If I made a tiny one, I'd get a golf ball and a tee. And the idea was, if you gave them constant saturating growth factors and optimal matrix underneath them, would the shape control it? And that was probably my most highly cited paper in Science, two Science papers, 94, 97, where we showed that cell shape does indeed, or cell distortion governs cell behavior. So that's how I got into microchip world, because the method we used to make those islands was a novel computer microchip manufacturing technique that George's group had developed. It was called soft lithography or microcontact printing. And so having done this, and George was starting to use these to make microfluidic systems where you have little hollow channels. You could think of like two channels that are like tributaries of a river, and they come together to make a big river. He was using those because due to laminar flow, there's no mixing, and you could do all kinds of interesting manipulations, and that's helped miniaturize all sorts of instrumentation. And I looked at that, and I had worked on vascular biology for years, and to me it looked like engineered microvascular networks. And so we started to culture cells in those in a straight way. And then and this is a true story, the postdoc that worked between us, Shu Takayama, went to University of Michigan, and I saw him 3-4 years later at a small meeting, and he presented what he called a lung on a chip, which was one of these microfluidic channels, no cells at the time, but he put a little fluid droplet through it, and he recorded the sound that it made, which is exactly the sound that Hans and I learned in medical school to listen for through a stethoscope, called the crackle. And when we were in medical school, that's, for you listen in the lungs with a stethoscope, and we always ask the professor, what is the crackle? What makes that sound? They go, I don't know, fluid on the lungs. And here he was showing it was like a particular size channel, the size of a bronchiole, with a fluid droplets going through. I thought it was great. And his student, Dan Hunt, applied to postdoc with me. And I remember sitting in my office and saying, that's really fantastic, but like, why don't we build like a real living lung on a chip? And the idea was to have a matrix at the interface between two flow channels, because George Whitesides had precipitated electrodes between two solutions at the interface between two flow paths. And we'd have lung epithelial on one side, lung capillary cells on the other. We'd recreate the alveolar capillary interface. And then also try to figure out how we could apply breathing motions because of my mechanical interest. And long story short, Dan, we tried that, it wasn't robust, and Dan came up with using a flexible porous membrane and little side chambers to apply suction that let us stretch and relax. And that was the beginning of organs on chips. And I want to emphasize that these are truly organs on chips, not tissue on chips, and not cells and microfluidics, which we and others had done before that, because it's really the tissue-tissue interface that is what's critical to go from tissue to organ. And that's where new functions emerge. And then the physical environment being critical, we have found consistently since then. But that's how I got to that. And I will finally say that we've explored IPS, we've explored primary cells, we've explored cell lines, but we have found the organoids are really the best. So I think we came to two places, but sort of ended up in the same place.
So did you ever manage to produce a crackle in your lungs?
I'm sure we did. We just never recreated it because we get all these fluid bubbles, but sometimes when it's getting into the airspace with mistakes, but he did.
Yeah.
He did.
Funny.
At least somebody did, right?
Yeah.
But it's amazing to see the convergence. It's amazing to see the overlap here as we're kind of starting here. But maybe to kind of see again some of these connections here, Don, if you may want to continue the conversation, as we're talking about the history and this convergence here, what have we really achieved thus far from your own opinion? What have been some of the greatest applications that have been enabled by these novel human models, this new way to approach academic research in human tissues?
I have to say, I'll talk about my world because I know Hans has found similar, is that we've been able to demonstrate clinical mimicry, basically replicating responses seen in human patients at levels that I never expected. You know what I mean? We did one study, we developed a cigarette smoking robot with like a Gatling gun of real cigarettes, and it mimics smoking parameters, and the smoke goes to the airspace, not an extract of the airway chips. And we did transcriptomics analysis, and we basically looked very similar to results of a clinical study of healthy patients who smoke cigarettes. The reviewers said, well, yours look much more similar to each other. There was much more variation with the others. And then we went back and we realized that we actually could do like that type of clinical study better than a clinical study because it was matched comparative modeling of the same patient before and after stimulus. Those transcriptomic changes were due to cigarette smoke exposure. Whereas those patients had different, work histories, family histories, smoking histories, and yet look pretty damn similar for, the top genes and the bottom genes, but there was also greater variation. So that was one quick example, but, we've done, we've modeled enormous numbers of disease processes. We've discovered new therapeutics on these chips, one of them based on breathing motions and seeing that breathing regulates viral infection. With influenza, that led to a licensing of that IP. That drug is now in trials for COVID because it prevents the cytokine storm, which is really the high morbidity part of the problem. We've done chips with cells from patients that create bone marrow chips that reconstitute white and red blood cell formation for a month. But I think one of the novel things that maybe is unique about organ chips versus the organoids is that we could, because we have flow, we could mimic drug pharmacokinetics so that changes in drug exposure profiles, drug levels over time that patients actually see. And this is something, as I think we're both MD PhDs, we know, clinically, drug pharmacokinetics are absolutely critical. They kill or make a clinical trial that, it could be toxic in one patient in kids versus adults if it with different pharmacokinetics. Yet in my whole career doing lots of drug work, I've always bathed cells in drugs, and that's the way most people do it. And what we could show consistently is that we could replicate drug regimen specific toxicities and responses when we mimic the drug exposure profiles on the chip. So that's something that I think we didn't think about in the beginning that is very, very powerful. We've also made those same chips with cells from kids that have a rare genetic disorder called Shwachman-Diamond syndrome, and we mimic the genetic disorder, which is cool, but we also uncovered insights into the etiology and mechanism behind the disease in terms of different phenotypes that no one ever knew before. So I think, we can go on and on, I'll let Hans speak, but these really are, I think, replicating human responses and have to be thought about as, replacing an animal study, not replacing a 96 wall plate from our side. And they're not as high throughput, but they're usually incredibly high content. And you could make them from old people, from young people, from men, from women. You could do things that you can't do in a clinical trial. Non-human primates are close to human, but they're not the same as human. FDA has funded us to make non-human primate lung chips, which we have done, and they look a lot like human chips with, let's say, viral infection in the lung, but they're also different, and so the things that you can do in these systems that you just can't do in animal studies and you can't do in clinical trials, like, we do gamma radiation exposure that you're never going to do in a clinical trial on these chips. So I'll stop there.
Yeah, so if we look back over the past 10, 12 years or so originally there were just a few labs active. Most bioengineers working with cell lines. Most iPS people were trying to make a single cell type out of iPS cells, like a neuron or a beta cell or something like that. And so initially, I think, maybe a handful of labs on both sides of the technology were essentially just developing protocols and trying to validate, disease models, come up with protocols to generate novel tissues or organs in a dish. Finding examples where they're predictive, where they give insights, and where they can replace existing other approaches. And that's what my lab is also, much like what Don was describing, has been doing, just basically coming up with protocols to study a particular cell type or tissue and then apply this to something that is of relevance. And I think one of the early successes in Holland was actually came out of our children's hospital in Utrecht, who had a big cystic fibrosis research group, large patient population. The Vertex drugs have just come out, magical drugs for these compounds, but developed for only a subset of these patients, about half. with a particular homozygous mutation, and all others now come in thousands of different flavors, all other patients, and essentially had no access to this drug because they'd never been in trial for that particular mutation. And the compound is really the way it works. It's actually two compounds then. Is really specific to that particular mutation for which it was developed. So this pediatrician walks into my office and he says, can we actually take tissue from my patients and set up a test in a petri dish based on organoids from these patients to score if this would actually work? Because then that gives me a reason to actually then give the then very novel drug, give it to my patients. And that turned out to be an extremely powerful black and white test that now, I think Holland has about 1,500 CF patients. The overwhelming majority now is in a biobank. And we don't grow airway organoids. You can actually grow airway organoids quite easily from sputum. But the most black and white tests turn out to be rectal tissues. And there's also the same gene product, the CFTR channel, is expressed and functional in rectal tissue. It's a painless biopsy. They grow very well. And there's no other channels that would sort of obfuscate what you're actually trying to score for the CFTR channel. And that's now basically a standard assay and gives the CF patients that don't fall in that 50% for which the drug was developed, but they still have access to this drug. And that's now actually being rolled out in other countries in Europe as well. So that was an early application. Another one that we immediately thought was going to be interesting. This is not so much organelle chips, but really organoids, that you could use these mini organs as a source for organ transplantation. And we originally, and this was sort of the immediate thing that as a stem cell biology think of course, it's probably by far the hardest to really get to work. But that was, so we worked together with a group in Tokyo led by Mamoru Watanabe, and we showed actually from a single gut stem cell grown in Utrecht, you could grow enough organoids to transplant it to mice. This is all mice. Transplanted to mice with IBD, an IBD-like set of symptoms in Tokyo. This worked well. And since then, they worked very hard. And so almost 10 years later, they now have their first patient in a trial where they're treating inflammatory bowel disease patients that are refractory to their treatment by taking biopsies from the healthy parts of the colon of the individual. And then 4 times a week, they grow it up tenfold every week, so 10,000 fold. There's a quite dramatic expansion of tissue in culture in the form of colon organoids. And then they're transplanted back onto these lesions in the hopes that, like what they did in the mice, they'll actually cover these lesions and break this cycle of chronic inflammation. So that was, but there in between, I mean, I learned from this whole process, we only saw it from the sidelines after the first study. It is very tedious to get something in an academic environment from something that looks great in a mouse to something that can actually is good enough to at least be tested in humans. So we're very hopeful to see that this works. Yeah, and then there are many other like COVID. People have used it for COVID. I think another great example that was done by multiple labs is using brain organoids when this Zika epidemic broke out in Brazil, and there was this coincidence of lots of babies born with microcephaly, with underdeveloped brains. There was an association, but no causal relationship. And adult brain neurons didn't really care about the virus when they got infected. But using these brain organoids, essentially that model the rapidly growing brain of a human fetus, it turned out there was a stage of these dividing neurons that was extremely sensitive to the virus. And that immediately explained why the brain doesn't grow out as it normally should have grown out in these little kids. So that was also quite an early application. Cancer is another one we can talk about later. But I think in essence now, the organoids will probably offer you any platform that we currently use, cell lines for or animals for, and then the organelle chip format will then allow this to be really standardized to measure very carefully to get multiple parameters out of a single bit of tissue.
Drew, if I could just make one other comment, one thing that to build other levels of complexity on top of this. One of I think, big novelties of the organ chips, because we have flow, we can grow complex microbiome in contact with human cells and for extended times, many days. And we did a cystic fibrosis chip with primary cells that look, very much like the patients in terms of ciliary frequency and mucus, excess mucus. But we could also grow Pseudomonas aeruginosa, which is in all of our lungs, but highly overgrows in these patients. And the patients that are living longer with these great vertex drug are still have morbidity and mortality due to Pseudomonas infection. And we could show that they grow many times faster on these chips from the patients with cystic fibrosis. We've now, with Gates Foundation funding, we've made intestine chips with microbiome from healthy kids versus kids with environmental enteric dysfunction, which are malnutrition in low resource nations. We literally get organoids from kids with intestinal organoids from kids in Pakistan, and we could compare them to healthy. We break the organoids up, put them on the chips, and we have endothelium on the other side. And we could show that while the organoids from these kids are a little bit different, they don't really look like what we see in the patients in vivo because we have clinical biopsy transcriptome. But if we expose them to malnutrition on the chip, only the organoids from the kids, the chips made from cells with organoids, the kids in Pakistan, look like in vivo. And now we put microbiome from those kids, and it even looks even more so. And so I think that's an area that's going to be really interesting. And then the other thing is, with inflammatory bowel disease, we now have organoids from IBD patients, which are epithelial on top of our layer of our chip. But we have the stroma from those patients. And we could do developmental biology experiments where we do tissue recombinance. So we could do, IBD epithelium with healthy stroma and vice versa. And it turns out the stroma is the major driver of inflammation. And this is really interesting because almost all the drugs target epithelium and immune cells, and now you have a whole new target area. And we could do exposed to carcinogens, and we see DNA mutations increase copy number only in the IBD chips. And so, this, I think, in counterpointing these different technologies, you get to a higher level of mimicry, and you could do experiments you cannot do on patients and you cannot do in animals. And so this is where it gets really exciting.
I think we come back to that shared North Star multiple times of the differentiation, the next level of what you can do with this work. I mean, the applications that you talked about, even in the last decade alone, have been so far reaching. I mean, Don, I think this is a really good parlay if you wanted just to start here quickly. While we're finishing our last kind of ground setting question here, if there's anything else you wanted to share about your work within the Wyss currently, your work has also been the engine to multiple companies and in the space, to the both of you that have kind of seen this and have been able to translate some of this work. And so I just wanted to ground set with our audience if there were any other applications you want to discuss or just chat with quickly.
Well, I mean, there's a lot of technology fallout whenever you develop a new technology. And so, I mean, part of our grant from DARPA in the beginning was to develop inline sensors. So we have all kinds of sensors for, transepithelial electrical resistance, for oxygen, for lactate. But we also developed, wanted to develop multiple cytokine detectors. And there you're dealing with very small volumes. And that led us to have to develop entire new technology using electrochemical sensors and we had to get the background down because electrochemical sensors are used in like the glucometer. They're cheap, they're mass producible, but they use an enzyme because if you do affinity, like with antibodies, affinity-based detection, there's a high background. So we developed a new nanocomposite coating that gets rid of that. You could sit it in plasma for a month and there's no background. That's now led to whole companies that are doing handheld multiplex diagnostics where you could do multiple ones in a matter of minutes in a handheld with disposable chips. So there's things that come out of this that you never expect, but Emulate is commercializing these around the world, but I think, it's a challenge, and we'll come to that in the future because you have to change the mind frame of people that don't want to, their job is not to take risk, right? And so to really change the way you do things, that's hard. So I'll leave that for later level questions.
Of course. And Hans, we'd be remiss to ask, I mean, you have several applications, but I think with Roche's commitment to the new space through the launch of the new Institute of Human Biology, I would love to hear more from your perspective and your own applications and forward within Roche and the Hubrics and many other sides on your side.
Yeah, maybe to explain a little. So I moved to Switzerland early last year. So I've been here for slightly more than a year. Before that, I was in the supervisory board of Roche. So I knew sort of from a high level what was happening in the different parts of Roche and Genentech and our companion in Japan, Chugai. And it was clear that organoid chip technology was sort of getting ready to enter the large pharma space. And there were multiple, I visited, I basically abused a little bit my position and visited many labs around the world in this company. And I could see people trying it out. And academia, I think, had meanwhile quite broadly embraced this new technology and started adding many new protocols and examples of how this could work. Of course, as Don already said, the industry is a bit more orthodox. Eventually, you have to pass whatever you want to do by the EMA or FDA. They, by definition, are very careful with new technologies. Everything needs to be extensively validated. So, but I felt that this would be a moment where this step could be taken to try to introduce this technology. If you think about it, you can essentially use it everywhere where we use live cells, be it animals or be it cell lines or be it yeast or zebrafish or something like that. And an early space would be safety toxicity. And as Don already mentioned, PKPD, can you build reliable, really human models that are robust and are strongly validated against the existing models and also against, I guess, clinical data that have been obtained with the samples. And that was happening already, but in a scattered way. So I felt this is a moment to step in. And actually, there was already an activity to build it kind of an institute in Basel in our big site here, in our R&D site. And that we turned into what we now call the Institute of Human Biology, which is hopefully going to bridge academia, where there's a lot of activity in this space, with industry that by definition is more reluctant to change what we have and what we know works by something that's more experimental. And also, I think it might be a place where, well, the regulatory authorities reach out, so they like to hear from large pharma companies now. Apparently, what is this technology? What do you think you can use it for? Will it complement? Will it replace, for instance, animal experiments and things like that? So that is actually quite exciting to see. And I think we're at the turning point that you also see it in academic literature, actually, that you can see people now manage to get the word organoid or organ on chip in their paper because it helps to publish it. And so even if you wouldn't really consider it to be an organoid, it might appear in the abstract or in the title of that paper. And I think it'll soon be the same thing if a biotech or a pharma company has some drug development program running, and they haven't somewhere implicated organ on chip, organoid technology. People will ask, why didn't you do this? Are you not missing some evidence here? And eventually, my personal conviction or my dream would be that we can replace many of the sort of 1.0 versions of models by these 2.0 models that really are closer to human physiology and human pathology.
I am so excited for how this conversation has been teed up, and I think we have a lot to discuss here. I want to pass it over to my colleague Chris to really take over our first section here. It's just covering human models and drug development. Chris, please take it away. I'm excited to dive in more.
Thank you, Drew, Don, Hans. I think what excites me most about our conversation today, and you've already started both of you to touch on this, are the differences in your perspectives, but the shared North Star of patient impact, especially when it comes to, as you were just saying, Hans, and as you've been saying all along, Don, modeling human physiology in such a way as allows us to really create and drive towards patient impact. And so you've given us a bit of that level setting about some of the technologies and some of the applications. And it feels like the holy grail and the overarching aim of this process and human modeling is to unlock the model to medicine approach. So Don, maybe you can start, but we'd love to ask from your perspective, where are we in terms of realizing this goal? Are we still early in the process? Are we a long ways away? Or maybe are we getting closer than we might expect?
Yeah, I mean, I think because I'm an MD PhD, I've not really trained as an engineer. I somehow become an engineer without realizing it. I've always used human pathophysiology as the benchmark. we weren't trying to build a model and show that cells produce, a lot of engineers who came into this in the beginning, it was like they'll do the minimum to show that the cells show some functionality, like a liver cell produces albumin, and whereas, we really wanted to mimic, the full physiology of these systems and the pathophysiology of systems. And I think in terms of the ultimate goal to me is drug development being expedited, faster, cheaper, safer, quicker. Whereas in the early part of the field, the emphasis more on toxicology. And it's interesting because it's much harder to convince companies to replace their toxicity testing, because that's where, they have the quick failure and people lose their careers. And I think it is easier to use this to show that you can actually do discovery of new targets, discovery of new therapeutics. I think that in the end is where the greatest value is going to lie. We've been doing this for a long time. So, we have multiple examples. I gave you one where we found that breathing motions affected viral infection, mapped out the signaling pathway. In that case, identified an existing drug, and we're quickly able to repurpose it. And then those data were included in IND. This is not a totally new IND because this drug had already been to phase 3, but and that is in clinical trials. And another one we were able to repurpose quickly, and by just looking for drugs that in COVID had worked against other related viruses. But more recently, we're combining artificial intelligence, machine learning approaches with transcriptomic data from patients, transcriptomic data from chips, and with that, we're able to really expedite the identification of existing drugs that now have functions you never expected. So repurpose. Every good drug is a dirty drug, right? No matter what anybody says, it hits multiple targets. And so, but more importantly, with using computational approaches, you can now map out the underlying networks and identify the targets that are actually responsible. We also have at the Wyss Institute, we have a broad range of know-how. So we have medicinal chemistry. We could develop new composition of matter that are even better than the other, but keep the good pharmacokinetic properties that are already known. That's a power of repurposing, that they're good oral drugs, for example. So I think you're going to see this sort of leveraging these systems to do new drug discovery. I think the other thing is personalized medicine, which, it's happening with the organoids in a big way, right? Because there you're getting from individual patients. We have a project with a clinician, a surgeon in McGill, and he has patients with esophageal cancer. And we've made esophageal cancer chips that have organoids of epithelium with stroma from the patients. And those patients get 3 or 4 drugs intravenously, all with a different time frame, so different pharmacokinetics. When you mimic that on chip and you give it through the interstitial space, the bottom channel, you mimic the patients that are resistant or sensitive. If you give it directly to the epithelial organoid-based channel, they just die. And so, I think this is going to be used to come up with optimizing drug regimens, drug frequencies, drug types, drug combinations. So I think, to me, the biggest areas are going to be drug discovery, insight into disease etiology, underlying mechanisms, new targets, and this sort of personalized area. And then the microbiome is like, there's no other way to really test these types of things. And the final thing I will say is that when it comes to toxicity and even efficacy testing, oftentimes people have to go to non-human primate. And what's happening right now is they're not available and they're incredibly expensive. And I'm seeing that as, and maybe Hans could comment from his side, but I'm seeing that as maybe one of the biggest forces that are pushing people to look in this direction who might not have before.
Yeah, so I can add in here that actually we see a lot of this, that they're no longer available in the numbers that the industry would need them. Of course, there's the ethical concerns. Everybody who works with them really doesn't like it very much, and still they are not as predictive as a real human model would be. So actually, we in the past years, particularly also there are particular drugs, particular antibodies, or things that affect the immune system that essentially cannot be modeled in rodents because the epitopes are not there, and also in monkeys for one or the other reason are not really predictive at all. So we've had about 6 or 7 programs for initial approval for the FDA for an IND. Where a large part of the evidence we put on the table, or in one case, all evidence, derived from in vitro human models, not per definition. So a large set of different things, some organal chips, some organoids, some essentially primary tissues. And that was accepted. And actually, FDA has been reaching out and said, well, maybe, we want to know more. Would there be a way by which we can interact a little bit and see, where this technology can help us out? I believe there's also a new law on the FDA now in the US that the FDA can no longer ask specifically for animal experiments, but that word is replaced by preclinical experiments, as I understand. And that, I think, it all points in a way that everybody knows that the system as we currently drive it is not sustainable, and that we will have to come up with alternatives.
Yeah, that's called the FDA Modernization Act, and it explicitly mentions organoids and organs on chips in it. And it was a big fight to get through, but in the end, both sides of Congress, Republicans and Democrats, supported it, which is unheard of in America. So that just tells you that the times they are changing. Yeah.
And as we think about those changing times, maybe let's pivot slightly and talk about those trends and predictions as we get your opinions on a few different areas through a quick rapid fire. So first and foremost, Don, then Hans, what do you think are the most common misconceptions within the application of novel model human organisms in biotech?
I think one, they don't think they're reproducible, and actually the reproducibility is better than animals. I think they think that they're just so oversimplified that they're not useful because they're trying to benchmark against a whole human, but you're really benchmarking against existing animal models, which is, terrible. So those are the two things that come up for me.
Yeah, I hear the exact same comments, and variability between individual organoid models, which actually organal chips already in part takes away, because you take an organoid of a random shape and you put it into a shape that you have defined, and in that sense standardized. So that's one thing. Another sort of interesting complaint I've heard: people who are used to work with rodents, for instance, or cell lines, the noise in their experiments is small, because every black 6 mouse is essentially the same mouse. And if you now do, instead of 10 black 6 mice, you do 10 humans on as an organoid, you'll find the noise of an outbred, genetically heterogeneous population. And so the typical pharmacology doesn't like that very much, because there will always be outliers. And if you just do a black 6 mouse or a BELC mouse or something like that, you don't get that trouble. Again, we would probably argue, well, maybe these organoids and these organ chips are better representations of who you eventually want to treat than this clonal expansion of a single mouse genome. Anyway.
I'm totally behind. That's exactly the one thing I was going to say. If you didn't cover it, is that we're not inbred, and that's why these drugs fail. That's why the same drug has different response in different breeds of mice, and then you go to human and they fail. It's crazy, and we want that variability. That is the power of these approaches. Because you could, I think, in the long run, you could basically get a small subgroup that are genetically related and make the organoids and organ chips from them, and use those very patients for a small trial, and that would increase the likelihood of success and shorten it and decrease cost. It's just changing the way you think about the problem, and it's hard when if you fail once, you lose your career. So people don't like to take these risks.
So maybe working backwards from that, then, when do you think we'll see the mindset shift that aligns with the rate of innovation happening in this novel human model space? Obviously, you're both doing the work to push this forward. And as Drew mentioned earlier, Don, with Emulate, Hans, with the new Institute of Human Biology, there are some really great drives to change minds and make it more commonplace. But would you have a sense of what might help drive that change?
I mean, the first thing I want to say is that, there's a difference between academics work in these areas and really changing the way drug development is done, because you have to get the same result with the same system wherever you are. So it means it has to be commercialized. It has to be mass manufactured. You have to get someone who's never used it before has to be able to get the same results as someone who's worked on it for years. And so in academia, people are always iterating, improving, and coming up with the system that's better. It may in fact be better, but if you can't get it to every company and they all get the same results, and the FDA sees that they can try it and get the same result, it's never going to be used to really translate into the drug development industry. So that's taking a long time. And then comes what we talked about before, people not wanting to take risks. But with all that, it's beginning to happen. I think Hans just gave an example. I gave some examples, but there are different drivers. But we published a paper in December where we showed that human liver chips made by Emulate, testing against 27 drugs, 870 chips, was 7 to 8 times better at predicting liver-induced injury. Now, that's cool, but what was more important part of that paper was that we did an economic analysis, and that saving, the economic impact of that finding was saving the whole pharmaceutical industry potentially $2 to $3 billion a year by preventing failure in late-stage clinical trials due to liver injury. I think economics is a driver, the non-human primate shortage and cost and ethical issues is a driver. And it's just like Hans said, it gets a life of its own. Once one company starts using these and they get in faster and they get approval and it's safer, then everybody's going to do it. And it's great to hear that Hans has got some of these INDs go through the FDA, because that's what you need.
So I think it just will take time. What I do see here, Roche is a very large pharma company with a reach that's worldwide in its R&D. So it's embracing the technology in many different places, but it's a different ecosystem, as Don says, from the academic world. And also, I think the way I've been developing in my lab in Utrecht organoids, so the organoids in Matrigel are the most flexible format, and every day you can do a different experiment, design a different way. The moment you want to standardize this, make it useful for drug development, that's when all these restrictions come in, that it has to be standardized, has to be robust, has to be cheap, has to be reproducible by a skilled, averagely skilled lab worker, etc., etc. So yeah, that's happening. Well, Amulet is a company. There's multiple other companies that pick up various aspects of this technology. So I'm not really sure that it needs a lot of push. I think it has now the kinetics of its own, the drive of its own, and will probably slowly take over.
You've both been true innovators of the human model space. Your seminal works have been the engines that have set the standard for organoid and organ-on-a-chip technologies, and you've also driven them both forward as well. As individuals who spent so much time innovating here, I'd love to take a moment to step back and ask, how do you think about approaching the human model space? At a high level, could you describe maybe your design thinking processes? Did you have any really strong guiding principles or philosophies, maybe a frame of mind that guided your approach?
Yeah, so I've always really been in the discovery space. So basically in that part of biology, mouse human biology, where very little is known. And what I've learned there over the years, the most, at least for us, the most productive way of working is set up a system in which you can reproducibly, you can use these changes and make reproducible observations, and then just observe. Don't hypothesize, because your hypotheses are just wild guesses. And once you have a formulated hypothesis, you are going to prove yourself right. And so I constantly find myself discouraging people in the lab to define a hypothesis. So you can ask a question, maybe, but not too deep, because your question is already driving you in a particular direction. So ask a very light question and then just observe. And then things will happen. You'll find deep principles, hopefully, if you keep your eyes open and keep an open mind. It's very hard because you immediately think you see patterns. You start creating these cause and effect relationships in your brain, and they're almost always wrong. And it's very hard to step away from that kind of thinking. And then a few times during my sort of 35, 40 years in the lab, you realize now I have a real new principle. And so the way we've exploited this, and one of them would be, when we learned how to take an adult stem cell and turn it into a mini organ, an organoid, and it was all done in the gut. That we then realized this can probably be done for many other organs. And basically, what we then the mode of the modus operandi of the lab changes. So basically, we then reach out to people who know about lung diseases and about the microbiome and about cancer and basically get these very active short-term collaborations and to essentially find as many applications as we could find in infectious disease, in hereditary disease, in cancer, in basic gene function analyses, et cetera, et cetera. And then work together with another lab in a discipline in which we were usually a novice, but then with the insights from that partner and our technological and I think Don, which you're describing, you're also, if I may say disrespectfully, all over the place. But as long as you have a collaborator who knows that particular discipline very well, and you bring in your technology, you can do unique things. So that is sort of another phase in the lab. So when there is some sort of a technical breakthrough or some insight that's deep, then we basically apply it very widely and then very rapidly try to find evidence this is true, this works. We can actually contribute to problems in other disciplines, et cetera, et cetera. So very opportunistic, I would say, how we have worked, but it works. And the hardest challenge is when you don't really know how something works, to not claim that you know and keep a mind that's as open as possible. And that's extremely hard for anyone, I think, and also for scientists.
So I start from a very different perspective, but end up, kind of similar. I started out as literally, a 19, 20-year-old with a hypothesis and a vision and intuitive insight about how biology works that, in a general sense was that mechanical forces are as important as chemical genes in many ways, and also that nature uses a very particular form of architecture called tensegrity. And those ideas drove me to get insights into how cancer works and extracellular matrix and mechanics, and that led me to then pursue how do I explore this, and if it's right, either it's going to be right or wrong, but how do I experimentally test those ideas? I had one reviewer who told me that this paper starts out with a hypothesis and goes about testing it and improving it, and he said it should be turned down. He should just show the data. And I said my 5th grade science teacher taught us hypothesis and educated guess, and science was then going about testing it. So it's a little bit different perspective, but I have been lucky in that my intuition turned out to be right. But what it did is it led me to do very much discovery work in the early days, but to develop new techniques to get at the questions I had. I developed pulling on integrins with magnets because there was nobody had mechanical tools in those times. And that led me, like you said, to reach out to engineers. And you learn by collaborating, and then to recruit students and postdocs. You learn from your students and fellows. And you begin to assimilate this like you're living in a foreign country and learning a language without realizing it. And so somehow, I got involved in tissue engineering in the 80s and formed one of the first companies because I was a biologist and the engineers wanted to do something, but they didn't know the biology. So you learned by this sort of just being open to following exciting ideas. Over time, in the last 15 years with the Wyss Institute, there's no doubt that I had done enough at the basic science discovery that I wanted to really have impact in the nearer term. And there, I think what I like to say we do here is not even focused on problem-based science, but solution-based. Like we're not only trying to, uncover what the problem is, but we're trying to think from the very beginning, like what would be the most effective solution. And we try to think about it not only scientifically, but economically. Can you commercialize it? Is this something you do with a startup, with a partner? And so, at the Institute, we've really created an Institute for Disruptive Innovation. And it's been quite amazing in that we're responsible. We're only 11 part-time faculty, core faculty, another 15, even less part-time, and we're responsible for more than 20% of all of Harvard University's IP and startups every year, We have something like 4,000 patents, 1,200 issued, billions of dollars in startup funds raised, over 55 startups, 120 licenses. So we created a model to actually to enable this sort of solution-based research and translation. And so that is, something that has affected how I now, once you start being successful, it becomes a positive feedback loop. Just like once you start collaborating with people outside your discipline, you see the excitement. You're not bored doing the same thing you've always been doing, and you keep growing like a snowball. And so it's sort of been an interesting process to watch.
So it's interesting what you say about solution oriented rather than problem oriented. I think this is typically what engineers are, right? I mean, I've learned this very recently. They know what the solution should look like, and then they develop it. They just go and develop it. Whereas we're always, no, we know what the problem looks like, and we hope to find the mechanism
Right.
behind the problem.
I've
And
been
it's
very successful at getting DARPA funding for the last, since 1998, continuously. And what they want is, my first program manager said, we're not interested in the mere difficult, only the impossible. And what you really have to do is write a science fiction short story about what you think the possible solution could be, and have some path to explore that. And, organ chips started, well, we started organ chips first, but we got a $37 million grant to build 10 different organ chips and an automated instrument to run them. And that was a purely impossible thing. I mean, purely impossible. And to be able to use it to quantitatively predict drug PK/PD. And we did it in 4 years, which was amazing, but it was a pure, science fiction short story. And we've done that again and again now, because once you do it, you realize that it pushes you past what you thought you could do. And that, to me, is what's now exciting about science for me. Not that I didn't love the discovery side of it as well, but to be able to have both sides of it is I find incredibly gratifying.
I'd like to say something more about my sort of my experience over the many years with collaborations, because I mean, as you also say, Don, this is really key to find trusted collaborators. And so what I've actually got from my PhD advisor was not a very visible scientist, but a fantastic mentor. And he always used the word trust and sharing. So he says we share everything with everyone, unless somebody disappoints you in abusing, and we trust yourself, trust your people in your own lab, in your own research group, but trust any collaborator. And actually, yeah, I encounter, well, in academia, there's a lot of, no, this is secret, you can't talk about it, I need to publish it first. That has, I mean, we've never done that, and I've actually never been in a situation where I regretted sharing information or reagents early. And actually that gets reciprocated. So if people know that you do this, 5 years later, you might be in a position where you need somebody to send you something or share something, and they'll do it because they trust you. And so that's the sharing and the trust. And I see this even more, this in industry, the sort of the secrecy that we have to adhere to, all the IP things. And I'm not really sure, and all the MTAs and the CDAs that need to be signed, I'm not really sure if it helps anyone, or if we would be a little bit lighter on this, if we would not be, of course, we don't want to give out, our secrets on an expensive drug we're developing, but everything earlier than that. And actually in the pharmaceutical sciences space, much to my surprise, there's a lot of interaction between pharma companies, because they're all in the same boat. They need to come up with assays that will predict safety, toxicity, PK/PD, and you'd rather together convince the regulatory authorities that something works and is a useful assay, rather than everybody has their own voice there, and the regulatory authority only gets confused and then says no to everything. So sharing, trust in collaborations always helps you.
I think that's fantastic. I never heard it quite put that way. I work with Judah Folkman as a postdoc, and he literally had a senior tech he paid full time, and most of her job was just culturing primary capillary endothelial cells and sending to people, anybody who asked them around the world, which built this entire field. You know what I mean? And I learned the same thing there. And I agree with you. I mean, at the Wyss Institute, we develop new models for collaboration is a big part of what we do, not only academic, but industrial. We have a a brain shuttle program that we developed that's a pre-competitive consortium, because every pharma biotech that wants to get, large molecules into the brain has the same challenge, but they have their own drugs that they own. And so we have them working together, and they're incredibly generous, open with the problems they've had, the targets they've pursued. And we've got 3 fantastic shuttles right now that are better than anything out there, and they now are licensing them non-exclusively. And I think we need more models to be explored to get the companies working together, because there's so much know-how. And you're right, in the end, each one is going to have their own drug, that they do own. But yeah, openness to collaboration is key. One thing I say, if you really want to do transdisciplinary, interdisciplinary, disruptive work, you basically find the most brilliant person you have who's passionate about a high value problem but can't solve it on their own. And then you bring equally brilliant, passionate people around who have the complementary skill set. And as long as they trust each other and get along, you get out of their way, and it happens, and it really works.
I think this is a perfect vision here on collaboration, especially as we're looking towards the future of the space. But Don, if we will, as a continuation, if we could do a verbal DARPA application here, more a speculation on the space, the future of the space. I think as we're kind of wrapping up here, and we've discussed much the potential impact human models are already generating in human health and disease. As we're closing up here, we'd love to just take some quick moments just to discuss the future of the space, really from both your perspectives. I mean, as both MD, PhDs yourselves and pioneers that have moved this space so far forward, just to start here with some quick insights, what does the world look like when we're able to completely integrate novel human model systems into medicine? What's the roadmap for success? When will we really know we've kind of quote unquote won in a way?
Yeah, so I think there's, as I already said, many different applications, in academic science and anywhere along the drug development pipeline. So I think one very visible thing might be personalized medicine. So testing medicines on patients' tissues before you test them on the patient themselves. This needs to be automated. This needs to be cheaper, faster. The machines need to be able to be run by a technician with a good set of skills, rather than a highly educated postdoc who spent 3 years to master the technology. This is happening. There are companies developing this. Yeah, I think where we will have to see a lot more development is in these more complex models. Don already mentioned microbiome in blood vessels in combination with epithelial cells, but particularly sort of immune-mediated phenomena. To just mention a few that I hear every day is the anti-drug antibodies. Large molecules, it's very hard to predict before you really are into clinical trials if your antibody will be immunogenic or not, and that will get in the way of a successful development of that drug. So that would be one. Building chronic inflammatory, the models for inflammatory diseases like inflammatory bowel disease, asthma, that sort of thing, where they're really, speaking from my immunology background, or multiple sclerosis, where the animal models, the mouse models, will have the symptoms, but they don't really have the disease. They have the symptoms because some manipulation that is not underlying human disease. So almost by definition, you'll find great symptomatic treatment, but you will not cure that disease. And there's that I think there's a huge unmet need. It's something very acutely important at the moment. Immune oncology drugs, extremely hard to predict which ones are the best ones unless you do a very large phase 3 trials. And you look at the statistics of many large pharma companies and biotechs who basically have to wait for the outcomes of their phase 3 trial to see if their checkpoint inhibitor is not so much safe, but actually has any effect on top of what we already do. And many of these trials just don't meet the endpoint, and there's no way to find out what is the right combination, what is the right drug to develop. So I think we'll see many more complicated models, particularly involving the immune system, the microbiome, maybe integration with elements of the nervous system. And gradually we'll get closer to the real human situation. the real human condition. But I think we should always be aware we are trying to simplify the human body and take it into individual elements. And eventually, no, a human body is more than the sum of its components. So eventually, I think clinical trials will always be necessary. But we might take away some of the less than optimal tools that we use on the way to go to phase 1, 2, 3 trials.
I totally agree that immunology, autoimmunity, complex immune response is absolutely critical. I mean, we've developed basically a lymph node chip that can very faithfully model vaccination responses, and we now have really exciting results with a Lyme disease model that basically is sort of a pathogen-induced autoimmune model, where you get hypergammaglobulinemia and T-cell suppression on these chips. And then the real advantage, I think, in many ways of the organ chips is that you can link different organs together to create human body on chips. And so, we did that with 10 different organs at one point, but it's an incredibly complex thing to do in terms of manpower and timing. But we did a first pass model that we were able to use computational PK/PD models, pharmacokinetic, pharmacodynamic models, to actually predict human pharmacokinetic parameters for 2 different drugs with a liver, kidney, and gut models connected. What that means is the potential to shortcut clinical trials design, but be able to figure out what dosing regimen to start with in a clinical trial. So that's the sort of thing I hope people will pursue to some degree. We've linked the lymph node chip to intestine chip and done mucosal vaccination, put in dendritic cells and so forth. I think you're going to see the organ chips come together in those sorts of multi-organ physiological, pathophysiological responses type modeling. I think that's some of the future. But, I always, and of course microbiome we talked about, but I always end my talk saying that the way big pharma often does it now is they'll do a huge clinical trial, costs tens of millions of dollars, if not more. They often fail, but what they do is they'll do statistical number crunching to see is there a genetic subpopulation who responded better, and if they're lucky, they find it, and they do a focused clinical trial with that group, and they get approved for a narrow application. I think with organoids, organs on chips, you could flip it on its head, and start with 50 of patients that are the same genetic same ethnic group and sex and age and genetic subgroup, and not only test for efficacy, but test for toxicity in their organ chips and organoids, and then use those patients for a small targeted clinical trial. And I truly think it would be faster, cheaper, you get safer drugs and more effective drugs. And if anybody ever does that, I think it's going to revolutionize the industry, because it's going to just save so much time and money. So I'm hoping that that's where the future lies. And personalized medicine, absolutely. And but I think the cost challenge is absolutely huge. You don't want it just being in the most expensive hospitals in the United States, right? And so how do you do that? I think this clinical trial is personalized, but it's personalized as a population. I think that's more approachable. Not to say that it will be used in expensive hospitals, with individual patients. And I think it's just going to change the way we think about drug development. I certainly hope it is.
Yeah. Actually, we have to think of sort of the workflow. We have done quite a bit of this for cancer, to grow tumors, test them, and then see if they predict what happened in the patient. But the workflow, the hardest part is not to have the pathologist throw the sample in formaldehyde before they give it to you, because that's what they typically do, right? And then they start thinking and sending out, and then you get a fixed piece of tissue which no longer grows as an organoid, unfortunately.
Yeah, that's another whole mind frame changing.
Yeah, no, so I learned, clinical workflows, if you can't fit this in, it will be very hard to implement it.
As well as who pays for it, and so these are big challenges.
Yeah. Completely. I think that was the one question I wanted to have around this clinical innovation aspect. There's so many things that need to change, but both as MD, PhDs, and kind of rounding out this conversation, I feel like we've chatted a little less. We've circled the topic around patient impact and how this will impact clinic over time. Just to quickly dive into this aspect as a bit of a closer or last question, how do you feel advancements in human models will really impact the clinic over time? And potentially even just how do you view this progression as technology progresses, as scale and throughput just increases along with cost?
So the hopes of, the way I've tried to sell our institute here in Roche is that actually it'll make, if everything is in place, which will take a number of years, it'll probably be faster, more efficient, and cheaper to come up with medicines that really treat the disease of a human, rather than something in an animal or in a cell line. So that's already a huge benefit for the patient. Wouldn't really notice, just gets a very good medicine that's maybe more available than the ones that we have been developing in the past. So, but I think the personalized medicine approach would be, if that, so I've seen it at work in cystic fibrosis, where the patient numbers are low, there's no hurry, but where it is shown you can actually perfectly match a patient with a drug that will actually help that very patient. And so that tells me that this can probably be done for cancer. And so then it would essentially mean that when you go and see your oncologist, the tissue biopsy is taken. Often now they're already sequenced. There's always histologies, of course, always all the scans that are being done, all the blood work. But it would be, I would anticipate it much like what we now do with bacterial infections. We essentially get the bacteria out of the patient, test them against a panel of antibiotics, and pick the best one and give that to the patient. In principle, we know this can be done with cancer samples. Logistics is not there yet, but the technology, on paper, you could draw out what kind of a machine, what kind of an instrument you would need for that, and this should be doable. And then, as I said, the workflow, you need to be able to somehow in the process, between the patient and the sample, that actually the sample is still alive and it reaches the diagnostics lab, rather than turn it into DNA or into a paraffin block.
I think that's a great analogy, because in sepsis, they give patients broad spectrum antibiotics, which have some effects, but every hour that they don't give them the specific, correctly targeted antibiotic, death rate goes up like 7 to 9%. And in cancer, we give them this generic cancer drug that has been tested against like 50,000 people, of which on average they could see 2 to 3 months increase, whereas some of those patients had a lot more better effects, some had none. And if you could do this sort of testing and also optimize the regimen, like how often you give it, because that might just be off for some people because their metabolisms. So, I think that's where the personalized power comes in, that could be truly transformative if we can get the costs down.
I know we have certainly gone over on a few of these topics, and I really appreciate the time and the dedication you both have had to this. Any closing thoughts you each would like to share? Anything you'd like to kind of have in finality as we're coming to a close here on the episode?
Let me just say that in the past 2 or 3 years, I've really become optimistic that this technology will contribute very significantly to patients' lives, to the drug development industry, and they will all benefit from this.
Yeah, I believe that's true. I think that the senior leadership companies know that this is coming, and I think some of the guys in the trenches know, but it's hard for individuals to take that chance individually. And so how it happens is going to be some successes, and then it's like I was told by a professor that science moves. It's like a tour group with an umbrella, and they're all following it. And all of a sudden there's a different color umbrella, and one person notices it, and two, and all of a sudden everybody's following the other umbrella. And it's going to be a tipping point. Somebody's going to have a success, and then it's going to be a wave. But it's going to happen. It's just it always takes more time than you think, and more money.
Well, I'm excited for the organoids flag to be waving along with novel model human systems. I think it's been amazing to level set and talk about the future of the space here from both your perspectives. We're just so grateful for your time. This was an amazing conversation. But Don and Hans, thank you so much for joining us again for a great episode.
Thanks for the opportunity.
Yeah, thanks, Drew and Chris, and thanks, Don, for being in this together.
Yeah, let's do it again. Bye, guys.
Bye-bye.